सेल में होने वाले मेयोसिस को समझाएंगे। हमारी क्लासेस ऑफलाइन और ऑनलाइन दोनों मोड में चलती है। सफलता के लिए जल्द से जल्द जुड़ें।
कथाएं बनती हैं। अर्धसूत्री विभाजन एक विशेष प्रकार का कोशिका विभाजन है जिससे गुणसूत्रों की संख्या आधी हो जाती है। इसमें 2 चरण होते हैं।
हमारी क्लासेस की सबसे बड़ी विशेषता यह है कि हम परीक्षा के पैटर्न और पिछले वर्षों के प्रश्नों के आधार पर पढ़ाते हैं। हमारी तैयारी पूरी तरह प्रतियोगी परीक्षाओं पर केंद्रित होती है। आज ही जुड़िए बायोशूटर।
Inside the nucleus, cell division operates as a highly choreographed molecular machine, moving far beyond the simplified sketches found in introductory biology. Success in the CSIR NET exam depends on identifying the specific proteins that dictate chromosome movement and shape. Meiosis functions as a mechanical assembly line where every physical shift is governed by a precise biochemical complex. Mastering the exam requires an exact understanding of which enzymes execute the DNA cuts, how they localize, and which structural proteins maintain the apparatus. Mastering these molecular actors provides the mechanical logic necessary to solve complex application questions. Prophase I begins with leptotene, where the cell must organize meters of tangled DNA so matching partners can find one another. Link complex proteins anchor the telomeres to the nuclear envelope and pull them into a cluster at one pole, creating the bouquet stage. Once the chromosomes are anchored, the SPO11 enzyme initiates programmed double-strand breaks, purposefully severing the DNA to begin the recombination process. During Zygotene, the cell utilizes these breaks to launch an active search for genetic homology. When a match is found, the SYCP1 protein assembles into a magenta zipper, mechanically locking the homologous chromosomes together to form the Synaptonemal Complex. This tight alignment allows one damaged DNA strand to invade its partner sequence, displacing a segment to form a D-loop. These early stages function as a precise alignment mechanism, ensuring each chromosome is locked to the correct partner before genetic exchange begins. The actual exchange of genetic material takes place during Pachytene. ZMM proteins dock along the synaptonemal complex at recombination nodules, marking the exact sites where crossovers will occur. The MLH1-MLH3 complex executes the resolution of these sites, swapping the DNA strands to establish a permanent physical connection. In Diplotene, the Synaptonemal Complex zipper disassembles and disappears. While the homologous chromosomes now repel each other, they remain pinned together by cohesin rings and the physical crossover points, or chiasmata. During diakinesis, the chromosomes hypercondense into thick, rigid rods to withstand the mechanical forces of the upcoming division. As the chromosomes push apart, the chiasmata slide toward the ends of the arms in a process known as terminalization. The entire Prophase I sequence exists to generate these chiasmata, which maintain the tension required for accurate separation. At metaphase I, bivalents align so that sister kinetochores face the same spindle pole, a configuration called monoorientation. This creates a mechanical requirement. The cell must separate homologous chromosomes while keeping sister chromatids joined at the center. The mechanical glue for this bond is meiotic cohesin, which utilizes a specialized glycan subunit called Rec8. To initiate separation, the APCC complex marks the inhibitor protein securin for destruction. Removing securin activates the enzyme separase. Separase cleaves the Rec8 rings located on the chromosome arms. This allows the arms to spring apart while the rings at the centromere stay intact. The central biochemical question is how separase distinguishes between the cohesin on the arms and the cohesin at the centromere. Protection is provided by the Zygotene protein, SGO1, which docks specifically at the pericentromeric region. SGO1 recruits the PP2A phosphatase complex to the centromere. PP2A then strips phosphate groups from the local Rec8 rings. Separase is structurally restricted. It can only cleave Rec8 when the protein is phosphorylated. Because PP2A maintains the centromeric Rec8 in a dephosphorylated state, separase cannot cut it, preserving the sister chromatid bond. Meiosis II follows, operating with the same mechanical logic as mitosis. The sister kinetochores now establish biorientation, facing opposite spindle poles. In anaphase II, the Zygotene PP2A protection is removed, allowing separase to cleave the remaining centromeric Rec8 and pull the sister chromatids apart. The mechanical distinction between reductional division in meiosis I and equational division in meiosis II relies entirely on the localized recruitment of PP2A by Zygotene. In mammals, these mechanisms are held in a state of dictyate arrest within oocytes in years or even decades. This arrest is maintained by high levels of cyclic AMP, which keep the pKa enzyme active. Active pKa inhibits CDC25B, which prevents the activation of the maturation-promoting factor, halting the cell cycle. This biochemical lock remains until an LH hormone surge triggers the resumption of meiosis. This extended pause leads to the physical weakening of the Rec8 cohesin rings over time. Premature degradation of cohesin results in loss of tension and the uneven separation of chromosomes during division. This mechanical failure is the primary cause of age-related aneuploidies, such as Down syndrome. The CSIR net frequently explores structural anomalies, including inversion heterozygotes. Chromosomes with inverted sequences must overcome a geometric hurdle to align homologous genes during Pachytene. To achieve proper gene-to-gene alignment, the inverted strand must fold back on itself to form an inversion loop. You must also account for species-specific exceptions, most notably in Drosophila males. These cells perform achiasmatic segregation, completing meiosis without the use of SPO11 synaptonemal complexes or recombination. Competitive exams specifically target these mechanical exceptions to test your understanding of the regulatory boundaries of the meiotic system. When all of these intricate molecular pathways function correctly, a single diploid cell successfully yields 4 distinct healthy haploid cells. Always distinguish between the reductional division of homologs in meiosis I and the equational division of sisters in meiosis II. For the CSIR net, focus your mastery on 5 key regulators: SPO11 for the initial cuts, Rec8 for the cohesin bond, SCO1 and PP2A for centromeric protection, and MLH1, MLH3 for crossover resolution. Solving high-level problems requires visualizing the physical localizations and active pathways of these molecules as they interact within the nucleus. Applying these first principles allows you to navigate the most challenging application questions on the exam.